Today, we're going to look at the characteristics and some components of adaptive immunity, and we're also going to develop an understanding of the mechanisms of adaptive immunity, recognition, and response to infection. Last lecture, we discussed innate immunity. We looked at the physical and chemical barriers to infection and the non adaptive rapid response to infection carried out by soluble molecules and our macrophages, neutrophils, and natural killer cells. Today, we're going to take a look at the adaptive immune system, which activates more slowly but results in a robust, highly specific response to infection that also results in the formation of memory cells that respond to any future exposure to that pathogen rapidly and with improved effectiveness. Today, we're going to be looking at helper T cells, which are involved in B cell activation, cytotoxic T cells, which recognize and kill self cells that have become infected or altered in some way, B cells, which are the cells that differentiate into plasma cells, which are dedicated antibody secreting cells of the immune system, and additionally, we're going to look at the role that macrophages and dendritic cells play in the adaptive immune system. Cells of the adaptive immune system specifically recognize a much larger number of pathogen structures when compared to the innate immune system cells. Additionally, after an initial exposure to these structures, the adaptive immune cells develop a greater affinity for recognizing and responding to these structures. B lymphocytes respond to the presence of both soluble and membrane-bound polysaccharides and proteins. And cytotoxic T cells respond to foreign proteins presented to them by infected cells. An antigen is a molecule that provokes a specific immune response. A single protein may have many different antigenic determinants or epitopes, and each can stimulate a distinct immune response. And antigen epitopes are what B and T cells respond to. B cells are formed and develop in the bone marrow. To keep B cells from reacting to self-antigens or the antigens of one's own cells, they're put through a positive selection process for self-reactivity. If you think about it, if B cells were to react to the body's own cells, that would trigger an autoimmune response, resulting in the destruction of one's own healthy cells. Now, a B cell has a structure on it that's very similar to an antibody. It's called a B cell receptor, or BCR. And this receptor is how B cells recognize foreign antigens. In the bone marrow, B cells are exposed to self-antigens by stromal cells, which are connective tissue cells like fibroblasts or osteoblasts. If a B cell binds to self-antigens during this process, it's then triggered to go through apoptosis. If the B cell BCR doesn't bind self-antigen, then the B cell can leave the bone marrow. A B cell has multiple BCRs on it, but they're all identical and bind the same epitope. However, the BCRs on one B cell will be different than the BCRs on a different B cell, which will bind a different epitope. In fact, it's estimated that a human has 10 billion unique B cells. When a foreign invader enters the body, if it isn't eliminated by the innate immune system, it will encounter a B cell that can bind to one of the many potential epitopes that it has on it. This process is referred to as clonal selection. Now, the idea here is that the B cell that binds the antigen is unique due to its BCR, and once it's fully activated, it will produce thousands of copies or clones of itself. But one more thing needs to happen first. Cytokines produced by effector T4 helper lymphocytes enable the B cell to rapidly proliferate. A single activated B cell can, within 7 days, give rise to approximately 4,000 antibody secreting cells, and over 2,000 antibody molecules can be produced per plasma cell per second for typically up to 4 to 5 days. So here we have that B cell that's bound antigen, in this case a bacteria, to its B cell receptors. Up above, a naive T helper cell is being activated by a professional antigen presenting cell, also known as an APC. So this is the APC here, and this is our T helper cell. The APC is presenting a protein epitope complex to a special receptor called a major histocompatibility complex, or MHC class II receptor, to the T cell's T cell receptor, or TCR. Now, a professional APC can be a dendritic cell, a macrophage, or even a B cell. The TCR binds or recognizes the protein epitope, and the T helper cell activates. Then, this now activated T helper cell carries out clonal expansion, making copies of itself. Now, back at our B cell, one of those T helper clones has found it. The B cell and the T helper cell bind via TCR and MHC class II. The T helper releases a cytokine called IL-4, and the B cell activates. Over the next few days, B cell clones are made, which differentiate into plasma cells, and these plasma cells produce enormous amounts of antibodies that bind selectively to the epitope and eradicate the infection. Also, during this entire process, T helper memory cells and B memory cells are also produced. These cells are already primed and ready to go to rapidly respond if this same affection occurs again in the future. So, what do antibodies do? They can cause agglutination, as seen here with these bacteria. They can also cause small molecules to precipitate. And, as seen up here, they can opsonize pathogens, which is the act of coating a pathogen to make it easier for phagocytes to grab onto them. And this works with antibodies because phagocytes have receptors on them that bind the stem end of monomer antibodies, allowing the phagocyte to more easily grab on to and phagocytose the pathogen. Another thing that antibodies do is neutralize pathogens. On the right, we see various mechanisms of antibodies disrupting viral activity by binding to the virus and interfering with the infection process. There are 5 different classes or isotypes of antibodies, also called immunoglobulins. IgM is the first type of antibody that is secreted by an activated B cell. It's a pentamer, which is to say that there are 5 individual immunoglobulin molecules, each shaped like the letter Y, attached together. Due to the pentamer shape, it's very effective at causing agglutination and precipitation reactions. And the first BCR that B cells present on the surface is a combination of a membrane-bound monomer version of IgM in combination with the IgD version of BCR pictured below. They both have the same affinity for the antigen, and IgD's role in immunity is not very well understood. It could have something to do with mitigating autoimmune effects. Now, using the monomer antibody IgG as an example, there are 2 variable regions of an antibody that can bind to antigen. The rest of the antibody is the same, no matter the antigen the antibody binds to, and the base of the Y is the portion that can be bound by phagocytes to their receptor to help them with phagocytosis. IgG is the improved version of antibodies that will have higher affinity for binding antigen that is produced later in the immune response, and also by memory cells if another instance of infection takes place. IgG can also cross the placenta in mammals, and thereby passive immunity can be passed on to a developing embryo or fetus. By the way, passive immunity is when an organism is directly given antibodies to bind an antigen without stimulating that organism's B cells. Active acquired immunity is the result of the process of stimulating an organism's own immune cells to produce antibodies, such as via infection or vaccination. Now, IgA is a dimer antibody that is found in body secretions, such as the mucosal regions of our epithelia. Additionally, IgA is found in breast milk, which introduces antibodies to the digestive tract of nursing young, which is another example of passive immunity. And finally, IgE is involved in triggering allergic reactions through the activation of basophils and mast cells. Now, since IgG can cross the placenta, there can be an issue with Rh-negative mothers who carry a developing baby that is Rh positive. Typically, this is not an issue during a first pregnancy, but can pose a problem for a second pregnancy. If the mother's immune system is sensitized against Rh positive blood, such as during the birth of a first child that happens to be Rh positive, in which blood exposure between the mother and baby happened during delivery, resulting in an immune response against Rh antigen. The resulting IgG antibodies that are produced from that exposure can persist in the mother's blood and cross the placenta during a second pregnancy, harming the fetus. Therefore, drugs such as RhoGAM, which is an antibody that binds Rh antigen, is used, which is administered to the mother to guard against an initial immune response to Rh antigen. If concentrations of this antibody are sufficient, the RhoGAM antibodies neutralize the Rh antigens of the baby's blood that contact the mother's blood during delivery. This keeps the mother's own B cells from encountering the antigen and triggering an immune response, which means her immune system does not produce anti-Rh IgG antibodies that can cross the placenta during subsequent pregnancies. Now, you might be wondering about A and B antigens and the effect antibodies to these might have on the embryo. Turns out the expression of these antigens comes later in development, and antibodies that target them do not negatively impact the fetus. Now, we've already talked a little bit about helper T cells, which recognize MHC class II proteins via their T cell receptor and are involved in activating B cells. Sometimes you'll see these referred to as CD4 cells as well, because they have cluster of differentiation 4 proteins on the cell surface. But now we're going to talk about a second type of T cell called the cytotoxic T cell, or TC cell. These will sometimes be called CD8 because they have CD8 proteins on their surface, and they recognize MHC class I proteins via their T cell receptor. And what cytotoxic T cells do is they initiate apoptosis in infected self cells via perforin and granzyme, similar to what we saw natural killer cells do in the innate immune system. The difference being how the cytotoxic T cells recognize self is via an MHC receptor versus the PAMP structures that we talked about in the previous lecture. Now, T cells also go through a self-reactive test, and this takes place in the thymus, which is where T cells mature. What happens is an antigen-presenting cell, such as a dendritic cell, interacts with T helper and T cytotoxic cells. If a T cell cannot bind the correct type of MHC receptor, or if it binds it too tightly, the T cell is destroyed via apoptosis. Now, cytotoxic T cells are activated by a professional APC, such as a dendritic cell, typically in the thymus. Once activated, the TC cell carries out clonal expansion, and the resulting cells leave the thymus to find altered self cells that present that same antigen. Once located, the T cytotoxic cell destroys the infected cell using perforin and granzyme. And memory T cytotoxic cells are also produced during clonal expansion. So today we looked at the fact that adaptive immunity is slow to activate. It's very specific in how it selects and interacts with antigens, and it also creates a memory response to combat infections that will take place in the future by the same pathogen. We talked about B cells, T helper cells, and T cytotoxic cells, and how they specifically recognize and respond to infection.